I'm very happy to introduce my friend and colleague, a guy named Doron Lancet, who I met on sabbatical in Rehovot, Israel. Doron was working at the Weizmann Institute, and I had the good luck to discover that Doron and I had a shared interest in self-assembled structures relevant to biology. My background is in membrane biophysics, and Doron's background is as a substantial computational aspect. And he had developed a kind of a software, or at least a program, that he called GUARD, and he's probably going to explain something to you about that during his talk. But basically, it had the idea that we don't necessarily need a long linear molecule to carry information from one generation to the next. Doron thinks that this is possible with assembled lipid structures, and this is now open to experimental testing. So Doron, I'm looking forward to your talk, and I'll turn it over to you.
So I thank, first of all, Noémie and what is it? Guillaume, that are hosting me in their home, which is unusual because of the state of mind of the UCSC, not allowing us to do this in the university. But I'm happy to do this here. They are very nice people. And thank you, Dave Diemer, for actually giving some introduction. Some of it is inexact, such as, for example, I'm not a computer sponsor. I am actually a chemist and a biologist and a computer scientist and an astrobiologist and so on. The only way to understand origin of life is to know as much as you can. So that's for students that want to do origin of life. Okay, so my model GUARD that I will describe to you.
Oh, guys. Really great meeting.
What is that?
Somebody has their...
Yeah, Clive, please mute your
Everybody, every mute except me.
Yeah.
So the model that I'm going to describe to you today is not computer model. It is a chemistry model. There is every tiny detail in it comes from the rules and the laws of chemistry. The computer is just a tool to find out what this predicts and how the chemical system in a test tube, in principle, can do the very first steps in the life origin. So what you see here, there are vesicles and lipid micelles also. They are our heroes here. There is something that you see at the bottom that is interesting in a sense that it shows how if you take something like a lipid micelle, which is a very small, the smallest assembly that there is in anything chemical that can be formed spontaneously. So lipids like each other, they form the micelles, but there are many different kinds of molecules in there. There can be a network all among them, and I will describe this much more in detail. One of the important things is that we start with... Which one?
You have to press this.
Press what? Is there any way of doing this here? Okay, so if it's there always, I will be okay. But touch it? No, that doesn't do it either. Yes, it does.
Yeah, touch it here.
Touch it here?
Yes.
Okay, and up and back
then,
here?
Yeah, and then if you want to...
How do you go back?
Like that. It's this
okay.
arrows.
Well, I hate this, but there is no other way.
Let me just check. Yeah, you can do the arrows, but not this one, but this two.
Yeah, okay, good, okay. So the idea is that life began with chemical chaos. And although some people think that's wrong, it's probably, in most likelihood, it is right. Because we get a lot of molecules falling upon us, and they make a very, very complex mixture of organic compounds in the prebiotic soup. You can see some comets coming and meteorites and dust that falls upon us all the time until today. and it always, some of that is organic. You don't have to worry about what to make life with. What is really important is how. So here is an example of the difference between how a lot of people see origin of life and how I do. On the left, you see Miller-Urey experiment from 70 years ago. And the idea was that if you take some gases, and there is a lot of argument about what gases were there 4 billion years ago, but if it has the right, if the gases are just right, if you pass an electric spark, you get amino acids. That's nice and well, but you don't have to worry about that because this comes as it is also from space. But some of it came from lightnings, which is what this is trying to exemplify. On the right-hand side, what you see is meteorite. Some meteorites are called carbonaceous meteorites. They contain a lot of organic compounds. And if you do very accurate mass spectrometry, as opposed to what Miller and Urey did, they just did some paper chromatography. They found one spot that they knew or found out was alanine, one of the 20 amino acids that we have in our body today. And they said, we found out how life began. This is as far from actual fact than can be, because just finding how with one way of organic supply, you get one of the molecules of life, this is not telling you how life began. Now, as for the meteorites, tens of thousands of molecules come down to us. And what you see at the bottom, you see these blue balls, they indicate amount inside the meteorite. And what you see in the X and Y, the Y is how many oxygens are identified in an individual molecule. And the right to left, the X is how many carbons. Something that has 15 carbons and 1 or 2 oxygens are in most likelihood something that we call lipids, or we call them amphiphils because the head likes water and the tail likes non-water, like oily stuff. And that's the story of the lipids. We will discuss it in a moment. But there is a very strong evidence for them being in a very large diversity. But experimentally, David Deamer showed this by exactly extracting, I don't know if the exact same meteorite, but probably like so. And what he found is something that makes vesicles, which means means lipids that can also make micelles. We do not attach to the idea, which we think is totally wrong, that life began at very first step, with those molecules that we have in life today. It could have been any molecule, but the principle is that at one point in time, Darwinian evolution has come in, and in that process, the Darwinian process, slowly there were selection events for what are the repertoires of what we have today. 20 amino acids, what we call the biological acids, sugars of different kinds, nucleotides like ATP that you see here, which is a monomer, a building block for RNA and DNA with some small modifications, and lipids. But what lipids? They don't need to be phospholipids. They don't need to be cholesterol. They don't need to be anything that we have today. This is after a lot of Darwinian evolution. So we stick with the idea that we have a lot of nameless molecules, but there is a principle that can lead them to life. What is the first stop in getting to life? It has to be multi-molecular entity. The idea that a single molecule, such RNA that makes copies of itself, which it never did in real experimental studies, 100, when you put it in water, will never make its second strand spontaneously, or you have to invent a lot of effort, which in what we call school chemistry. You have just to have this molecule, you have to have just the molecules, the small molecules that makes the second strand. This is not to be happening in the chaos of the soup the prebiotic soup or primordial soup. So from there, it is more likely that life began with a multimolecular entity, as you see on the right. Perhaps it will make a vesicle, but if not, it will make a micelle, and if not, some other ones will make that. So by the end result is that there is an entity with a lot of molecules, and they can easily talk to each other because these are fluid, both micelles and bilayers. Micelles and bilayers that surround vesicles, they are very, very fluid. So molecules can go from one place to another, interact with each other, generate a network of interactions, and this is the basis of early life. But how do we get that? How can we get to the point in which we have something like that that will self-reproduce and have babies that are similar to itself? We'll get to it in a moment. Most people look at life today, the origin of life today, as follows. Habitable world, that's important, that an astrobiological indication that the temperature is right, the atmosphere is right, the water is there, without water there is no life, and so on and so forth. And then they say prebiotic synthesis, mostly from gases. Until today, a lot of people work on how from very simple gases. This is methane, water, ammonia, H2, HCN, hydrogen cyanide, and so on and so forth, and they will make the bigger molecules. Some may have been made this way. Many of them come in ready to go. But that's just a small point. And then they move and say, the only thing that can make copies of itself is RNA and DNA, and we say it cannot. If they are alone. In a network, such as a living cell, yes. But starting from complex molecules, such as polymers that have 100, 200, 1,000 monomers, elements inside that chain, as a beginning of life, a prerequisite for life, we think that's utterly wrong. And we think that life began with small molecules, such as we'll put together micelles and vesicles, and also small molecules that can be inside the vesicles. There is water inside, as you see here. Can I make a? Do you know how to activate the pointer?
The pointer is here, so if you just move
Yeah,
it
but
to-
there is a way. If you do right side, you can get. I think you can get in PowerPoint a laser pointer,
Oh,
which is
that
like.
I never used,
How do you?
We can see your cursor on Zoom if you just.
Yeah, but it's very difficult for me to use the cursor. But anyway, I will try to do that.
Wait,
Okay, fine. So right here inside there can be, here it shows in all times, in all the papers, they show that there is RNA inside. It shouldn't be there. Small molecules will start, and 100 million years later, maybe RNA is formed. That is our part of our view of how life began. It's quite sure why you get so many question marks here, because nobody has ever made a protocell, as we call it. A protocell is a vesicle with some molecules inside and also inserted into the membrane or associated inside and outside on the surface of the membrane. All of these together have to reproduce to go the very long way to what is known as LUCA, last universal common ancestor, which is basically, if you ask how complex it was very like what we call today bacteria. So it had DNA, there were enzymes, very complex enzymes that help it make copies of itself, the DNA. Also, an enzyme that can make possibility to make RNA out of DNA, to copy the information from DNA to RNA. The ribozyme that can make proteins is a machine that reads triplets of monomers of single parts on the RNA to instruct making proteins. And those proteins can be good in just the right sequence that will allow them to fold and make tiny machines that we call folded proteins. They can be enzymes, they can be all sorts of other things. So all of that is missing at this point, and there is a lot of question marks on the way to here. Can we solve the problem? What can be reproduced? I already heard the idea that RNA can reproduce. Not very likely. If people ask at the end, I will explain more why. Collected, that means that it accretes from the soup, protocell, for it to reproduce. No question that it cannot happen. It has to start by a lot of trial and error, and just random ones are very likely not. Most of them will not reproduce. What I will show that there is a route to show that a certain percentage of the randomly made assemblies can reproduce to begin with. Let's go there. First of all, we said, let's forget about the very large volume of 10 to the minus 6 larger than my cell that is called a vesicle with its content inside, because it's much less easy to do any dream of reproduction, copying yourself, because each of the molecules in principle should interact with several others in the assembly, and they should be with exactly the right way that will give rise, immediately to be said in more detail, to an assembly that can make copies of itself. Why micelles? Okay, being small is very important. So first is a single phase, no lumen, the water inside, versus the molecular structure of the membrane on the outside to reproduce exactly the same time and same timing. So synchronization is needed, but in a single phase, it's much less difficult. It's not that it is not needed. It is needed, but it is very easy to get, or relatively easy to get. Promiscuous formation. Most of the lipids do either vesicles or micelles. And if you have a very large number of types, some of them for sure will do. Some of them don't, but that's fine. We are thinking Darwin. Whoever fails goes away. Whoever survives will do the rest. So promiscuity is very nice because a lot of different types of molecules in the mixture can make these structures. Energetic downhill formation. Why do they want to be together? And as you see in the picture, the tails that don't like water stick in the middle and adhere to each other, and that's downhill when you are in water. No details right now why. And the head groups like water, and they're very happy to be on the outside. So everything is just perfect. And that's what we call in chemistry downhill. This means delta G of Gibbs, negative, and that means that there is either increase of entropy, of disorder, or if it is going down, which is what happens when you make a micelle, there is something else called an enthalpy. And if the enthalpy is negative enough, the delta G is negative and it goes downhill, which means, in simple words, it happens. A chemical reaction that can take place. Undergoes growth and fission. This I will show here. This is a 30 years old paper from Luigi and others. And what happens is that you can, don't want to go into all the details. Let's only look at the right-hand side. I may get to the left side as well. So the right-hand side is that they can grow by just accreting more of the greeny ones. Mostly, and Dave Dimmer tells me why, most people, experimentalists, do only one type of molecule at a time. So that's what Luigi did. He took one of them. This is actually a fatty acid. And the fatty acid, the green, does the micellation, and it then accretes more and more until it gets a little bit unstable, as you see in this, something that looks like it's about to split. And then it actually splits. And the fission gives two progeny. Okay? So this is quite easy to understand, and it has been a lot of evidence. Okay, so just a second. Just a second. Okay. So we get convinced that it can undergo growth and fission. The growth and fission is extremely important, but what I will show you to say how life began focuses on the growth. You'll find out why in a few minutes. Established catalytic reactors. If I step here, there are hundreds of papers, mostly from the industry, that show that micelles are an unbelievably good arena for doing catalysis. This means that one molecule can enhance the rate of one or two molecules or more than molecules that undergo reaction together. The original first molecule is a catalyst, and it enhances the speed of the chemical reaction. And you can see this all sorts of examples. I won't go into it, but you can see on the right, the middle one from top to bottom is just one amino acid, proline. And it can actually, because of the micellar environment, no details right now, it can be so that a single very small molecule can be a catalyst for quite a complex reaction that you see at the top of this middle square. Okay, and that is true for all the other reactions. So micelles are very, very good at generating a network of oops, sorry, it does it goes here. Okay, sorry. Okay, so the idea is that it is a wonderful catalytic reactors. Much less is a vesicle for many reasons. High concentrations, fluidity. The reaction, if you look at the reactions, and when two lipids, for example, have to interact with each other, the concentrations are about 1 molar. This is unbelievably large. People always say, where do the micelles can come from? If you are already, or what do the lipids, how they can talk to each other? The answer is, if you're in a micelle, because of the very small dimensions, high concentrations, also fluidity, molecules can get in, can get out, and also interact with each other non-covalently as well as covalently, namely by weak and strong chemical bonding. Mixed polarities, the surface is very polar, the inside is very apolar, and there is a whole gradient which is extremely important for catalysis. And then easy transition to vesicles. I will say just one thing, and that is that it can go from micelles to vesicles spontaneously, and therefore we can continue. So this was an experiment. I'm not telling you all the details because there is no time. So let me stop for a moment. If there is a very crucial question until now, please ask it by talking. Don't be shy. You have to open chat and then chat if you want to do that. Okay, never mind. You wanted to activate your microphone, but if you don't want to do that, I'll continue. So this is just a different look at what life started with. In fall, you see this, whatever it's a comet or something like that, coming into Earth with all of the organics. You don't have to synthesize. Info is mostly enough. And then micelles can begin the job because they spontaneously form and they can also grow and split. But we have to do something very important, and that is explain how you actually get reproduction. When RNA replicates or reproduces, it copies the sequence of the nucleotides in the RNA. What would be something that is similar to that will come up immediately. There is no sequence here because all is very fluid, but what you can do is do something else immediately to be said. So this is like non-covalent polymers that replace the covalent polymers for the very early steps of life's origin. And then the micelles can be real protocells because if they can be reproduced and they undergo a very small change that will make them the same phase of lipid will be then surrounding a protocell, it will bequeath the capacity to copy itself. It's not easy to understand, but the computer simulations show this in much exactitude. And then we say there is a potentially Darwinian evolution all the way from the micelles to Luca. And just like we don't ask how nematodes, very small worms, can make human beings along Darwinian evolution, not exactly right because, well, yes, maybe it is right. Anyway, a very small animal can become a very big one or a plant and so on. If that can happen by Darwinian evolution, we think that the micelles all the way to what we call Luca can undergo the route just by Darwinian evolution. That's our theory. but it has very strong legs in reality, feet in reality. If you want to copy something, some information has to move from one place to another. And the question is, what is the type of information? So it's very clear that for RNA, it's sequence, although composition, because there is A, T, G, and C, A, T, G, and U in RNA, and doesn't matter, 4 different kinds of chemical letters. And if what happens then, you can copy at least the composition, but definitely better the sequence, then we say RNA has made its own copies. But there is no sequence whatsoever, because the neighborhood of molecules to each other is fluidly changing all the time. So there is something else. What's that something else? The answer is, what is that? How do I get rid of this? Just touch here. Yeah, okay, no, but this also moved it, so I have to go back. Here, yeah, okay, thank you. You see on the right, something that we all know about for proteins, for RNAs, for DNA, sequence. What is near to each other in a string? On the left side, order is not important. Structure is not important. The only thing that matters is how many of each kind. You have 5 reds, you have 3 greens, and so on and so forth. That's composition. It turns out that compositional information is the most important part in cells today, but most people don't realize that. And this is what you see here. All of you that are biologists don't get in, I don't have to tell more about than that, but for the astrobiologists and so on, I will say the following. In the cytoplasm, the fluid inside a living cell today, there are many messenger RNAs floating which encode proteins. Each of them has its own quantity. And actually, we very often describe a cell by how many of each kind, irrespective of what the sequence of the RNA is. That's compositional. Same is true for proteins. They have functions. They have quantities. We don't care about exactly at this point in time. We talk about reproduction. All we need to know is that the ratios among the different proteins will play right. And if so, the life can continue. The growing cell will have the same proteome, as we call it, and the baby is also the same, okay, after fission. And the same is true for metabolic composition, small molecules. If you have too much of one, too little of another, death is almost promised. The only way is to make somehow, through a network, a system in which the composition will be copied. So what we say here, without in chemical formalisms yet, you have small molecules, those are the lipids, they make spontaneously micelles, and then this can grow more and more, as already mentioned, and you can get random fission. It turns out that the only important stuff here is what happens in the growth, and it should be homeostatic growth. What does it mean? That if you have 3 yellows, you get 6 yellows on the right, on the middle, more exactly. If you have 8 greens, there should be 16. And then when it splits, it will be more or less the same, the 2 random fission products, but they will be very similar to the original father before growth. That is how life began, we say. Very small, spontaneous, molecules are abound, and despite that, there can be a process in which you get copying from father to sons, or from mother to daughters, or any other way. There is no sex yet, no gender, but you have to give it to the babies. So the selection, the split is easy, the fission is statistical, but the homeostatic growth requires much more than that. It has to describe a network of reactions that actually fulfills this idea. And the father of how this can happen is Stuart Kaufman. And he came out with the following idea. Suppose you have a lot of molecules, never mind what they are. For him, it was BBAABB, which is peptides, namely small proteins, with only two types of molecules, A and B. But they can make a geometrically increasing number of types if it becomes longer and longer. So you have a lot of them. And if you have a collection of those, made all from the foodstuff A and B that is seen in the center, what happens next is that there is a point in time, where you have enough diversity and enough collection, the right collection of molecules in an ensemble. If each of them can be produced, for example, by a reaction that I show where is that? Okay, it's right there. So for example, if you take this reaction here, which is shown as a black square, it's A plus AB giving AAB. But the arrow in red is that molecule BAAAA can catalyze this reaction. Not only that, enzymes today, and the older ones, the precursors of those, let's say short peptides, can do the following. It can actually somehow recognize that the only two partners that should be involved in the reaction is A and AAB and nothing else. And also, if you just take A and AAB, it may happen very slow. Sorry, A and AAB, yes. No, sorry, again. If you take A and AB, and if you are just in solution without any neighbors, everything can be very slowly happening or not at all sometimes. But if there is a catalyst, then it happens. So it gets a network of catalytic interactions. And we say that's how life began. But what happens here is that if everybody in the ensemble, each of that has a catalyst that can help it happen from smaller molecules, be synthesized from smaller molecules, then there is some kind of a catalytic closure, as he calls it. And he calls that a collectively autocatalytic set. Autocatalytic because the entire set is autocatalytic, namely copies itself with rate enhancement events. And he calls this CAS, okay? And this can reach catalytic closure with certain conditions that were told by Kaufman, not always in the right way, but it led to some of what we say today. There is some set of conditions in which what you see here will lead to self-reproduction. So now we get equations. I'm not going to explain that, but please forgive me for saying that you don't, in as much as I can tell, if you can write the numbers in those equations, and I will say a few words about that immediately, and in the equations are known for many years to be right and describe what happens in catalytic reactions, then if we show by simulations by computer without any experiment that we get self-reproduction, it must happen. And it also is in principle very easy to find by experiment. But what you do when you do equations like this, it tells you exactly how to do it with experiments. I will say just one sentence. The technology today and also the computing of today cannot immediately show that I'm right, but it's going to happen very soon. One example of how to decide about numbers is the following. Here I will go into only one detail. You see the beta ij in red on the thick red arrow. This means that green helps blue to come in catalytically. We have a number that says by what factor it makes it speedier. The answer is it's 5. Or it's 237 times, and so on. How do I put in a matrix that includes all the numbers that are necessary to say how every color can help any other, including itself, to come in? And the answer is that it is a matrix, and we put the numbers that are very strongly related to very clear experimentally based set of numbers that people find. What happens when you have a lot of such numbers and you have to put it right? It has to be log normal. Whoever understands what that means, it's okay, but it actually is something that looks like this curve, okay? Very many at very low beta IJ. So most of them are very small catalytic effects, but at the tail, like the IQ tail, very few are genius, but very few are very strongly catalyzing. And we can say what is a fraction of that. And that means that I did not do my figment of imagination wasn't what puts the numbers there, but actual, well-described, experimentalized kind of set of numbers.
What are the experiments?
Which ones? For the one before? Okay, so to get this curve, it's actually very simple. Maybe I would say it in a few words. The idea is that if you do the experiment, which you can do, a protein, just one folded protein, and 100,000 drug candidates, you can people very often find out if one or several among the 100,000 bind to the target protein. This means it is a good beginning for the drug development. Now, what they do is they measure the capacity to bind between the protein and every one of the 100,000. They measure that experimentally. And you can get the distribution of that. And this is it. If you change that, it's not right. It's not what you find in a real experiment. So what we ask and we get answer for is that, as you see on the left, in the center, you see what I said before. The speed should be proportional, the speed of entry should be proportional to what you already have there. Therefore, we will double everything and you'll have homeostatic growth. And then the split I already mentioned. Now, not every composition will do it. So if you make 2 trillion micelles, that's very easy to do on Earth. In a small little pond, warm pond, that Darwin has mentioned in one of his letters to a friend, Hooker. So he said that it's very clear that you have to have what you need, what can give rise to the kind of Darwinian evolution that we know happens today. You have to have something that will make more copies of itself, and it can happen in a small pond or very large oceans. It doesn't matter. There is lipids everywhere because meteorites don't care where to fall. So the idea is that you have the materials and what you have, and you cannot change the rules of chemistry. So the only thing you can do is make a lot of different compositions. Okay, so we call that very rare composition a composome. It's a composition, it's not the material molecules, it is the status. If it is in a composome, it reproduces. Let me skip this for a moment because there is not enough time. And this is just way, I will not tell you why this is so, but if you see a big red square, this means that you have time from 0 to 100 at the x and also from 0 to 100 at the vertical, from top to bottom. And what you see, if there is a square in the middle, it means that, say, points from 30 to 60 almost did not change their composition. This means there were many. Each of these numbers is growth split. So it means that you end up with a composome. And it can change. There are more than one. And you can see a transition right here at this point where mutations in the composition gives rise to another composome. Let me also skip on that and just say one important thing. This square, you also see if you decide that the molecules are chiral. What does it mean, chiral? You may know, many of you know, so excuse me for explaining, but the idea is that many molecules, most organic molecules are such that they have at least one carbon. You know, carbon can bind to 4 other atoms of different kinds. This quaternary story of 4 can be that 2 at least will be the same and 2 others are each of them, so 3 types of atoms. But if it's 4 types, there is a problem of symmetry because of the geometry of the bonds between the central and the peripheral 4 atoms. And this gives rise to 2 mirror images that are called chiral isomers. Okay, so the idea is that they are not the same molecule, but they have a lot of capacities which are very much the same melting group, melting point, or if it's a liquid boiling point, and all sorts of electric, many physical descriptions. Okay, but the 2 molecules are different when it comes to recognize each other. And it turns out that if instead of taking 100 molecules of different kinds in our simulations, we put 50 that are what we call the right hand and another 50 left hand, the composomes will be with selecting sometimes for more D than L or more L than D, which is something that people are trying to understand how it happened for many years. So this kind of formalism also tells you how you can get what is called homochirality. All amino acids are left, all the sugars are right in life today. Let me also skip here a little bit. I will just say that this is just that it is support for one molecule helps another molecule come into the membrane or into the micelle. And also that micelles can form vesicles quite easily, because if you change just a little bit the geometrical formation of the lipid, it will make micelles, if it looks like the triangle, it will be worm-like when you see in the middle, and it will be a vesicle at the bottom. So this means that it's quite clear that very small changes in the molecules will give rise to going from micelle to a much bigger vesicle. And I don't know if I have time to say all of that, but I will just say that just as there can be establishment of networks for entry and exit, today in our own cells, there can be proteins that help some molecules to come in or go out, and not many others. These are called transporters. There can also be pumps that can bring molecules against their gradient. Open 101 biology and find out, or biochemistry. So the idea is that you can actually do the job of not only entry and exit like transporters, but also like enzymes that make new molecules by making the strong bond that is between these two or between those two, which is not like what happens here. They are not bonded together, but this here has a strong bond. This is what makes what we call metabolism. And this is the beginning of metabolism. And it is also the first step to make RNA in the maybe 200 million years later. I will not tell you exactly why I think it's so long, but it is going to be very difficult to get. But it will happen with Darwinian evolution. So you can begin to have vesicles, and you can have some molecules inside if the reaction is that you have a head group like this one, which has 2 heads, so to speak. So one of them can break down and flip first and then go down, and one of them, the yellow, say go in. It can begin to feed what is inside, but all under the control of a mutually catalytic network. So everybody all along will make copies of itself. Some of them. Others will die out. That's usually happening in Darwinian evolution. I'm skipping all of that just to say that there is a very strong experiment by Stephen Fletcher, in which he utilizes micellar in experiment, a micellar system, with 3 different kinds. In this case, the 3 different tails are different. They have different colors in this picture. And there is 1 type of head, but there are 3 different types of lipids. And what happens is that on the right-hand side, without going into details, it goes into a flow cell experiment. And what happens is that you can show for a certain time, just like in the red square that I showed you before, that the relationship in counts or concentrations between the 3 colors that represent 3 different lipid types, that this will actually be time independent mostly. And this is basically showing composome in a test tube. Life today is based on proteins, and proteins are long. They can be several thousand amino acids. They can be only 50, like insulin, but basically they are very long. They are much longer than you can do in the following, what you see here. This is actually what we see in bacteria today, but it is a mimic in some respect of what Kaufman predicted. And that is, you can get, let me show you what we are seeing here. All of these are enzymes. Let's consider it enzymes, okay? And where they are stuck. And this is beginning of a short and then becoming a long molecule, which is only an oligopeptide, meaning several amino acids threaded covalently to each other. How do you make this that it will keep the just exact sequence that you need? The answer is that there is an enzyme, in this case of non-ribosomal peptide synthesis, that is experimentally known because it's in a bacterium. For example, this enzymatic complex here should take this one and to add another monomer here, but it has exactly like I told before, this specific 5 have to stick with another one, but only that typical exact one. And what we say is that the enzyme has to be very complex to do that. So it's something like 3, 4, 5, 6, this went fine, okay? Maybe also beforehand, except that the enzymes were not enzymes, but short peptides, but I already showed you that that is a possibility. But much later on, something unbelievable happened, and that is the advent in sort of some kind of talking to each other all the time. You have to get RNA, you have to get ribosome, you have to get tRNAs that are those that stick a single amino acid to the tRNA that then knows how to touch part of a trimer in the RNA, and this helps the ribosome, this unbelievable machine, to make new peptides that can be basically as long as you wish. This couldn't have happened prebiotically, and this couldn't have happened very early on, like maybe after 100 million years. But it ended up in evolution to happen because we all have it today. Let me skip that too. When we look at the composome, which is the bottom network, and compare it to the top one, what you see is that the bottom one is very small. Each of the circles is a different kind of molecule, and it has a serial number, but the size says how much. Again, composition. And what happens is that we see that when we go from, if you do random accretion into a network, it will, maybe I should say that by showing this, okay? You see on the left side, there is a meandering in a three-dimensional space in which represents 100-fold dimensional space of the different types. A vector, in this case, is the composition. The numbers are how much of each kind. It starts, let's say, in the black, and it begins to go through the red and the orange and the yellow, and it's continually changing its composition. That's not a composome, no reproduction. But at one point in time, it finds itself by we thought was random search, it gets to the composome, but that's not right. If you look at the right, we were able to show, again by computing, but using all the exact chemical equations that we have used before. And what you see here, all of these positions here, like here, the beginning of that, and the beginning of this, and the beginning of that, all of them are very far from composome. But there is something that we call dynamic attractor, and this is known to many physicists. Astrobiologists will probably know it much better than I. So what I'm saying is that no matter where you start, you end up with an attractor. And this attractor, you can't even believe that this happens. That is the composome. So there is much better situation, much better probability, both because of one thing that I will say again immediately, and the idea that there is an attractor, that you end up in an attractor and there is reproduction. One very important thing that you see here is that the composome has many fewer molecule types than what it started with. Why is that important? I said that there should be a reduction of numbers of different molecules. You start with 10 billion ones in the ocean, and you end up with a few thousand small molecules in us, and quite a few, but only in tens of thousands, not much more, polymers. So the idea is that this process of reaching the composome also does this reduction of repertoire, and that's extremely important. So if it happens many, many times, all along evolution, this will lead to this unbelievable going from billions of different molecules to many, many fewer. Yes.
Yeah, I have a question.
When you have a question, just shout, because I look here, so I don't see you.
Okay, the top one can have both chirality, but the low entropy state, the one that is reproducing, will only reproduce one chirality, right?
No.
How does it work?
A question comes from a lady that actually is very interested in how you get the homochirality. This is Noémie, and she's now a very good friend for me. I just met her today. And the idea is as follows. First of all, getting all the amino acids and all the sugars is really a very final part of the story. But we asked something very simple. Can you, if you look at a small network, will they all be exactly the same amount of DNL? Lipids are also, they are also chiral. And also the head group can be a ribose, the head group can be a peptide, an amino acid, and so on and so forth. So, okay. The question is whether it's all exactly the same amount of DNL, which is what you do when you do physical separation, or not. If not, then the central large number 61 can contain out of 100, maybe it has 30 molecules, but some of them, 10 are L and 20 are D. This is the beginning. It doesn't have to be pure, but the whole idea that there is a process that is continuously happening, and it is clearly chemical and well-defined, and I hope also supporting quite well with experiments. The idea is that you can get a beginning for what we call chiral chain, how do you call it? Chiral selection, okay? That one is more than the other. But this happens all the time. When you have an enzyme today, all the enzymes that to them approaches a molecule that has to be broken down, and it is D or L, one of them will get cut and the other one will not be. So in biology, that's very clear that molecules know if it's D or L. Okay, so this is based on that. Okay, I'm getting very near to the end, so that's not so bad. Here is one interesting story. We are going back 4.5 billion years ago, and there is ocean. There is argument about when this happened. I don't care. It only happened 3.85 billion years ago. It's just as well to me. When there is ocean, when there is infall, when there is a soup, when you get to begin micelles, something extremely important happens. I computed in the back of an envelope. This is another pile of enzymes that makes mu, but it's okay. So the idea is that you can make easily 10 to the 33 micelles on the globe. Now you say they are probably all the same. No, they are not. If you do combinatorics, if you know how many types of colors, so to speak, of lipids we have, you can get easily to the idea that you can more than 10 to the 33 different types of micelles. So this means that every single micelle is different. I think Darwin will be very happy to see that, because some of them don't ever copy their composition, but quite a few, maybe something like 10 to the 33 divided by 10 to the 20. If it's a very rare idea to get a composome, it is still will be with 10 to the 13 micelles around. Okay, so this is very, very encouraging, and we like this finding very much. It cannot happen with vesicles because when they are very big, then they are all the same. So you have to be very small. That is another reason, we already mentioned that before, that if you are very small, it's very good. So this is how life began. I want to see refuting this, not telling me, why don't you get to the cellar of Dave Dimmer in his home, which is unbelievably wonderful laboratory, and begin to do experiments. I will not. I will tell you at the last slide why not. But well, that it's difficult and it's unnecessary. Immediately will be told. But what happens is the following. I already said, you start with small micelle, it grows up, it becomes, as we saw from Luigi Luisi, it becomes unstable, it splits. And if you have the process I've showed you before, perhaps not the first cycle, but the second or third or fifth or 20 or 200, it will get, apparently, because of the attractor, and because there are so many to begin, if they all begin together, then it will happen. And then it goes further with what we call the GARD model, the graded autocatalytic replication domain model. And this actually begins to make covalent bonds. So you can begin to have short peptides, you can have short oligonucleotides, and they can grow, and they become more and more interesting and more functional and more bringing you near to LUCA. A lot of people say, well, but how will it ever become a vesicle? So I already gave you the answer. It's very easy. So I will not tell you more. Just change of chemistry will lead it to become vesicle. And if it's a very, very small vesicle, then micelles can be coming inside by fusion with the vesicle. And if some of them bring a new catalytic capacity, this will be good for the vesicle. And if it doesn't, tough luck. Darwin will say that. So this began Darwin. We get to this vesicle, and then it goes in some papers, not in all. In some people say LUCA begins. LUCA is the beginning of life. This is, I cannot find the right four-letter word to say how that's wrong, because LUCA cannot found in an ocean spontaneously. So it has to go this Darwinian evolution. And then, as you see here, this is the Darwinian evolution, what you see in the gray. The rest is LUCA going into the three parts of life today: eubacteria, eukarya, and archaea. Eubacteria, we usually call them bacteria. Anyway, so the idea is that there are many, many, many steps, Darwinian steps before LUCA, and this is how life began. So you decide when you call origin. I call the origin the first micelle. Some people will say the first vesicle. Some will say the first ones that have good transporters that control what comes in and comes out or get stuck in the membrane and so on and so forth. So this is in the eyes of the beholder. I wouldn't risk myself saying when on this diagram life began, but for sure life emerged. So it is an example that you see at the bottom. We have micelles, then small micelle vesicles, then those that have transporters. It can be just holes, or it can be with the protein that causes the hole and controls some entry or exit and so on. And then you begin to get oligomers. I also show that in the talk, because once you can do a dimer, you can do a trimer, you can do also a pentamer, but you cannot do much more unless you have RNA. So now you have to wait. The yellow arrow between E and F is extremely long. I don't know how, but it should go from very small number and different kinds of number, or maybe big number, but none of them is very functional. Some of them are functional. You can get my predictions can go through because there is self-reproduction. So also between E and F. But now you end up with something like RNA and ribosomes and cytoskeleton, DNA, and enzymes such as the proton ATPase. For those that know, you know that it is a very complex enzyme. Those that don't, it's a very complex enzyme that can also has a rotor, so there is even movement within the protein, and it actually passes out. I can't remember. It passes protons upon about feeding upon ATP, the energy molecule, but it can also make the energy molecule because of the gradient across the membrane of protons, which is H+, the hydrogen cation. So that should have taken a long time, and even more time, or no, maybe little time, because if you have already all of those components, Luca is very soon at the gate. So that's it. All I have to say is that I did all the things that I should have done. Last slide.
I think you got out of it.
I completely got out of it.
Yes.
Oh my God.
End of the beginning. Oh,
Yeah, no, but I have to have one.
so there is another talk?
No, that's this.
No, I'm kidding.
No, it's the end of the talk about the beginning of life. Can you still hear me? Can people hear me?
Yes.
Yes, thank you. There are always some brave volunteers to do that. Thank you very much. So the idea is futuristic, which is almost now today. There is this paper that made a prediction that in 2012, he said that, or they said that in 25 years, we will be able to do something that is now called molecular dynamics, to actually see how enzymes fold. We already have that. AlphaFold from Google. Protein folding, same. Accurate simulations for all of that. And then going perhaps in another 12 years to the entire bacterium. If this can happen, 100 molecules in a micelle will be very easy to follow in something that I know some experimentalists will not agree to that, but most people today think that something like molecular dynamics is almost exactly like doing an experiment, because we first do 100 experiments and see if it happens right in the computer, and then we do another 100 and we trust that because of that. This is how science goes. So now it's the end of the beginning.
Okay. Okay.
So open some microphones and ask questions.
Maybe I will stop the sharing. People can see you. And yes.
Okay.
I'd be happy to ask a question, but maybe we should start with an early career. Right, somebody.
No. Why don't you start? The early career always want people with old career to start.
I don't know if that's true.
In my lectures, always.
Well, I'm a late career who's largely ignorant of this topic, so my questions will be very simple, but it impresses me, this prediction of computational, being able to compute all of these probabilities and networks and understand in a predict.
Not the probabilities, the actual workout. What happens? Which touches to which helps another to get in? Everything, the entire chemistry.
Yeah, exactly. So it's predictive, but it's also probabilistic in that you're computing these affinities or these reaction rates and affinities to assess that or to quantify that, correct? My basic question is, if you can do that on a computer and solve this problem for Earth, can you change the conditions and solve this for Titan or any other planet system that has completely different conditions? Why not? And if not, why not?
I say, why do you ask? Because I am not making any decisions on what are the molecules. All molecules behave the same.
So are you saying that this is deterministic, that this is going to happen on any planet that has water?
In this grandiose way, yes. Tell me what can be, you are an expert on that. Give me a landing on extrasolar planet. You find that there is life there. Or want to find out if there is life. What do you think can be different in that particular faraway planet?
Well, you talked about hydrophilic molecules and hydrophobic molecules.
All molecules are the same unless you go.
So what if there's a different
If you go across the well-known boundary of between universes, which I don't think happens, if you cannot cross, all the laws are the same. Only the conditions can be different. The initial states.
Absolutely, but on a moon like Titan, you might have ethane instead of water, for example.
Okay, okay, thank you. That's very important. It has to be, I said that, but not it's strong enough. It has to be in water. If it's not in water, and if it's not the organic chemical that we know, but we know that on all of even our own solar system planets, we have, when you say ethane, you mean 2 carbons and 6 hydrogens, just like here. Okay, so it's the same molecule. If it's the same molecules, the same water, everything will be just right. You can say maybe the temperature is too high. So it will be potentially somewhat different process, but it will happen, most likely.
Yeah.
Is there anything else that you can come up with of what could be different in another planet? Because having them ethane or something like that, that's not a difference. We also have ethane here.
Surface pressure, or where the life is forming. Pressure might be pressure, temperature, and the liquid that exists.
That's an interesting question that can be solved in a moment. Try any reaction on Earth at 100 bar and see what happens. The constants change, just like the activity constants change, but the question is whether they will change very strongly. I think you should continue with work of finding more extrasolar planets, because there is a good chance that you will find life there.
Thank you.
Thank you. It's okay for people that just didn't understand anything to ask me questions, okay? Because
I have a question.
Thanks.
Can you hear me?
Yes.
Yeah,
Loud.
so kind of a follow-up to what Natalie was saying. My impression is that with like Earth, like very important to have some sort of abiotic like motor that can create like a kinetic trap, like in a freshwater example, that might be a cycling pool.
Go back, please. I don't understand what you're talking about. Please start with a question, not with what you know. Tell me what you are asking, please. I'm sorry about saying that.
What I'm asking is that, have you considered, on the topic of like what planets are able to produce life, have you considered the like necessity for some sort of like gradient or terrain that can produce some sort of like plausible like kinetic trap? You're just saying you could have like what
is a kinetic trap? I don't understand.
It's like some situation where you have synthesis like exceeding hydrolysis for the formation of these micelles or lipid vesicles. Like for like a freshwater origin, I've heard that you'd have these little cycling pools where there would be an accumulation of organics from cosmic bombardment.
I understand. I will give the answer. Life began with life, not with the environment, not with how much salt there is in the ocean. It has to be water. Now, you are talking about whether or not a particular reaction will take place. Will it actually have a negative delta G? This can happen when you do heat and cooling, or you heating and cooling, or drying and wetting. This is all very nice. But life did not begin on that, because the idea is that there are a lot of different reactions that can happen to each of the molecules. And if it does not have the right type of molecule, it will not work. So, for example, what we do by drying and wetting, or wetting and drying, the end result is making some molecules that will not happen spontaneously. Somehow, they get a kinetic trap. You're absolutely right. But the point is that if the molecule, like you remember that I had this purple and yellow, if they are not activated, such as with a phosphate or with some element of partition or of participation of sulfur and so on, like thioester and so on, if that is not there, the reaction will not happen. You can do the job of doing heating and cooling and get it happening. But the best way is to find a molecule around that when binding, and there should be like that, such as a pyrophosphate, which is on almost every volcano. These are molecules that when they stick a phosphate, say in an example, on a carboxyl, this is very high energy. And then it will make the peptide bond without heating and cooling. The whole idea that life will have to depend, or the origin of life, the very early steps, will have to depend on something like somebody out there, you and somebody else, heating and cooling, or maybe waiting until your planet will go around from night to day and so on. You cannot begin life like that. Life is chemistry. I will tell you more if we want to talk about it later. Thank you.
Hi, Professor. Can you hear
Hi,
me?
how are you?
Hi, I'm well. Thank you. Good to
We
see
met
you.
this lady yesterday in an airplane. And the first time that it happened to me, that after 1 hour flight, she decided to listen to my talk.
Yes, it was very intriguing. So I got a preview for today. I was wondering a very simple question. What makes each lipid different from each other?
What makes what?
What makes each lipid
Oh,
different
no.
from each other? So
Oh,
you,
no,
in
no,
your
no, no.
slides, you had like red and green ones.
No, hold on. Okay. They are not all different. My saying that there are quantitations means that you can have 67 of one and 42 of other. There are many, many copies. There is no question about that. There are zillions of copies all around you. What we have, what is singular is the composition. When you make the number 1 micelle, then number 2, number 3, number 4, number 10 to the 20, maybe they each will be different because of combinatorics. Each of each kind. You start with each of each kind, or a very small number of each kind, and most the others don't get into. If you have 2,000 outside, only 100 or 200 can go in. You end up with something that is very unique in composition. And then it begins its long journey to become capable of reproduction. Did I answer this, or do you still have a question mark?
I was just wondering, like each individual lipid with the head and the tail, is there a difference between any two lipids that allows the reaction of one to influence the other?
Yes, that's a very good question. The answer is the head groups can be very different. They can be charged differently. They can have be peptides, different lengths, different made of any of the 2,000 amino acids that are possible in the beginning, because there are many, many more than 20 that we have today, and so on. So you can have a very, very large variety to choose from of the head groups, and the same is true for the tails. Then can be changed by length. They can be changed with how many double bonds they have. Sometimes they are branched, even today. All of what I said is even today in life. So you can have, we calculated sometimes that it's a really, really big number, that you can have all of these combinations. This head with this tail, yes, you can have a lot of different lipids. Is that answering?
Yes, thank you so much. And
Thank
to bring
you.
up briefly what you said yesterday, it's very amazing that life exists much in the way that it's as if a Boeing airplane was created out of thin air, that we're here today. So really great. Thank you so much, Professor. I have to go now, but thank
No,
you.
don't go down. Don't go down for 1 minute. Yeah, now you can go down.
Okay.
There's a question from Alexander.
Okay, let's take Alexander.
Hello. So thank you for the really interesting talk. My question is about the composomes. So I'm sort of curious, like, how many different composomes you've found? Like, what are the composomes? Like, what are like the ratio of certain elements that are autocatalytic and self-reproducing, and like, do they match the composition of life forms that we know about? And also, sort of, how quickly do the composomes come to dominate the soup? Like, are there like one composome that would like cover the entire ocean, or are there like 10 different composomes like in a balance in over the whole world? So those are kind of my questions about the composomes.
This is very nice. Okay, so the idea is as follows. If you start from 10 to the 33, there is no chance that there will be a a bottleneck of one. Okay, so there must have been a lot of different entities all around. If you're a composome of the very primitive kind, namely the micellar situation, then you will get potentially in some kind of a pond or ocean or wherever, they will begin to reproduce and they will get a species, a group of micelles that are relatively similar to each other. And some others will be completely different, somewhere a meter away or whatever, or a millimeter away because it's very small. So the idea is that, yes, you get a lot of different kinds. In our simulations, a typical simulation gets something like 2 or 3 composomes, but they can't come back again and again because it's a very simple situation, just coming in and coming out of molecules. But when you begin to make new molecules, then sky is the limit. You can get a lot of different new molecules. So you get a lot of more diversity and you can get a lot of composomes. But asking how many composomes there are, and by the way, we call composomes up to a point. I assume that at one point in time, if we fly into space and find a place where we have something like a 1 micron vesicle with a lot of organic chemistry inside and have a whole colonial group that sits somewhere and they are all quite the same, you will call it life. And I wouldn't call it composome anymore, but it is in some respect also a composome. What was the second question? I think you had one more question.
Yeah, I mean, I think you answered a couple of them
No,
at once.
the answer is supposed to say what you are. Okay. Yeah.
My question, I think, is there isn't an answer because I was thinking like there are like 1 or 2 special composomes that reproduce and then take over, and the answer is there are many, and so they're not necessarily dominated by one composition.
No, not even Luca. Luca is shown in own pictures as if there is only one species. That's wrong. And there is a very strong movement in the APSICON, the Astrobiology Conference of Convention. That place, there were several lectures that said it is impossible that there was only one Luca, that all of us descend from it. There may have been 10 billion or 10 trillion all over. But every now and then there was a Darwinian invention, if I may call it that, that was so strong that they devoured everybody else. So this is a bottleneck. But you look at the trees that we see in all the books and all the papers. What does it look like? There are animals, and as you go in the tree, you get progeny in terms of speciation that are a little bit different, and so on and so forth, and so on. So it's very difficult to know how many lived then and how many were together, were contemporary. I would say, don't worry about this. Look at the tree, think about species, and call the first thing that I call species is a micelle with a specific composition. Because if it's a composome, there will be many more of it, right? Or a few more of it, more than one, which if you had the right machinery, we will be able to detect. Are there some neighbors that are very similar to you? And so on. So it's experimentally predictable, but not yet. And the idea is we will not know, but don't worry, it will come here. It's sort of funny to say that. Okay.
Thank you for the helpful explanation.
I know we're really over time, but can I ask one more question? So you're not proposing that—
For me, there is no overtime.
Okay.
Oh, really?
You're not proposing that composomes or the emergence of these composomes is a replacement for like RNA world. I mean, I imagine that self-replication of RNA is another thing, abiotic invention that has to happen concurrently.
No, don't continue. Don't continue. It's there. There was RNA in the next to last sphere that I showed.
I saw that.
Means that
so you're getting RNA into the vesicle after you form it.
No, it's not getting into. This is it minds blowing. I mean, it's not made outside. You cannot make RNA that is functional unless you are in a reproducing entity that Now let me say it in a clear way. If you go from RNA long 5 to long 6, and it is something that is capable of making some other molecules in the Kaufman picture, then you win and you continue. And if you do something wrong, you put the wrong 6th or 5th or whatever nucleotide, then it will die. Maybe because maybe it's poisonous. So the idea is that you test every tiny step to be right for survival. That's happening all the time with Darwinian evolution. Okay, so it's not that you say something like, you have to move from a kangaroo to a deer or something like that. That somebody places in a new mechanism for growing inside you the baby. It is happening step by step inside the animal. So nobody will come with a syringe of RNA and put it in. It has to be coming step by step.
Mm-hmm.
That's the way I see it. I had a very long discussion on this with Dave Dimmer today. We didn't get complete agreement, but I hope that he understands a bit more nowadays. No? Okay. So what would you say? How does it tell you something about extrasolar planets? Or does it?
I thought you were asking Dave Deemer a question.
No, Dave, I did not ask. Okay, so I just mentioned the name. Okay, go ahead, please. Is that changes your mind with respect to what happens in other place? Okay.
No, I mean, you ask what is the origin of life, and I see it as a continuous process from the Big Bang to us. I can't pinpoint an origin. I mean, I could say the triple alpha reaction that created carbon, or you said the origin of vesicles, or the origin of enzymes.
That's interesting philosophy. But it's very clear that you need to have all the elements. So forget about the supernovae.
Yeah.
It's very clear that you need water. It's very clear that you have some organic molecules here. So you have to have a habitable place as well as the right some amounts of what is necessary to start the story. So that's where life begins. But once you have the lipids, very shortly afterwards, very short time, it will begin to be somewhat alive. Okay?
I'm not going to debate that. I guess the question I would ask you, I mean, for me as an astrophysicist, this all does seem very deterministic. Even the choice of water seems very deterministic. But when I ask biologists, okay, you have your vesicle, you have nucleotides, you're assembling some complex chemistry inside of these vesicles. But then my question is, all right.
But not just putting in. It's all
forming inside. I understand.
Some catalytic effect.
I understand.
It's extremely important. Thank you. Go ahead.
Yes, I understand. So then my question becomes, all right, how many ways are there to encode genetic information? I mean, is all life expected? Is it so deterministic that we expect all life to be DNA based, for example?
I would say that none of what is written in the last sphere before Luca is deterministic. That it will be that type of unbelievable mechanism in which there is one polymer made of one type of monomers will be able, with the help of something that is unbelievably complex called the ribosome, which is also a ribosome, and that it will actually be able to use 3 and not 2 and not 4 and not completely other letters and so on and so forth. All of that, we don't even know that this is the only solution. Actually, maybe it would be nice to think, does it look unique, or can you invent? Some of the people with high imagination, maybe they would be able to design an idea that is completely different from RNA, ribosome, tRNA, and tRNA loaders.
Mhm.
Maybe that can happen.
Yeah, okay. I mean, this is the answer that I usually get from the chemists and the biologists.
Okay, so
And
what
at that point, but you're saying vesicles first, RNA, assemble the RNA through some catalysis inside the vesicle, and then who knows how the information is.
no. It's not assemble the RNA all along and then wait for the ribosome. No, it cannot happen like that. They should talk to each other. A
Yeah.
very super primitive ribosome with super primitive RNA. Then slightly less so with the counterpart, and so on and so forth. Same is true for the tRNAs. Same is true for what loads the tRNA. They should all co-evolve. Otherwise, it will not work.
Mhm. Thank you.
So there was never a point in which there was high length RNA that was waiting out there and said, tell me when to come in. That's not the story, I don't think.
Very interesting. Thank you so much.
Thank you. Very much so. Anybody else? Don't worry. I will not say hard words except if necessary.
Oh,
Shall we
okay.
stop?
Yeah, if there are no
Okay.
other
Goodbye, everybody.
questions. So
Thank you for
for
listening.
joining.
Thank you for the questions.